Here's a recap of some of the concepts in 8.3 photosynthesis for high level. So let's remind everybody where we are in a cell. Of course, we're only talking about autotrophic cells, and none of the reactions occurring out here in the cytoplasm, which is different to respiration. Everything's taking place inside the chloroplast. So we're going in there, and then we've got our thylakoid membranes here, a whole stack of them. It's called a granum, or grana plural. And we have here our stroma, which is our kind of liquid, kind of the equivalent of the cytoplasm inside this particular organelle in the chloroplast, where some of our reactions are taking place. Now notice that this is stroma with an R. So stroma, the liquid, and if you see stoma or stomata in the context of plant science, they are the little pores for gas exchange in the leaf. So different thing. And we talk about these membranes being thylakoids, and we talk about this thylakoid space, these gaps inside them as well. And here we have an electron micrograph of the same information. And these little black dots and little ribosomes to make the proteins, particularly rubisco, that really important enzyme in the Calvin cycle. So the chloroplast has 70 S ribosomes and its own DNA capable of give instructions to make its own proteins, which is an important part of endosymbiotic theory as well. Here's another image here, just showing again those same components. All right, so our overview. Inside our plant cell, inside our chloroplast, we have two main stages. We have our light-dependent stage here, and we have our light-independent stage, or Calvin cycle, or carbon fixation. So the first stage here, the most important things that are happening, the photolysis of water, so splitting water. From that we get hydrogen and electrons that need this carrier, this NADP, which becomes NADPH when it picks up the hydrogen. That carries our hydrogen ions and our electrons off to the Calvin cycle. And then our water is actually a waste, oxygen, is a waste product. That will just diffuse back out of the chloroplast and out of the cell, and then be exchanged just passively through those little pores, those little stomata in the leaf. And then our other thing that's happening here, we have photophosphorylation of ATP. So we're using the energy from light to put together ADP, an inorganic phosphate, to make ATP. So we're sending an energy-rich molecule and a loaded carrier with hydrogen and electrons into the Calvin cycle. That's the main purpose of our light-dependent stage. Now, if we look at that in a little bit more detail, we need to talk about our chlorophyll being our pigment. There's also some other accessory pigments as well. So things like our carotenes and xanthophylls, those sort of orangey yellow sort of coloured pigments, but we generally just talk about it all collectively as chlorophyll. Now that's contained in proteins that are embedded in this thylakoid membrane here, in this granum. And we can see here we actually have several molecules, and in the middle is the reaction centre. This whole thing together here, a collection of these pigments and these proteins, is called a photosystem. Our light energy, we just mentioned before, the word we need to use is photolysis, photo for light, lysis for splitting. That splits our water molecules. So we get from H2O, we get hydrogen ions, the H+, as well as their associated electrons. So if we have the nucleus of a hydrogen, so we've got one proton in the middle, and then we have one electron, and they separate so that we get a separate electron and our proton, which is our H+. And then, of course, our O2 gas that diffuses out. So these things are coming out of this first stage. And the other thing that light energy is used to do is to eject an electron from here, from the center of this photosystem. So an electron leaves because it's absorbed energy from the light incoming here. And we say that it's photoactivated at that point. So let's add another layer of detail here. Now we've mentioned here these photosystems, but let's have a look at how they fit together. So here, this is like one little section of our granum where we've got one fold of thylakoid membrane, and we're focusing on just 2 of these photosystems for now. So we've got lots of little chlorophyll pigments or other accessory pigments in our here, making our photosystem, and we've got a little reaction center in the middle. So we can see that some photosystems, they send their excited electrons into an electron transport chain, which is very similar to cell respiration. So here, lights come in, hit the photosystem, given it energy, and an electron has come off, and that's being passed along these carrier molecules here. Now, that is losing energy as it goes. It started out quite excited, and it's decreased in energy, but the energy has been used to pump hydrogen ions into here. So this space fills with lots of hydrogen ions so that we get the concentration gradient. So that's one thing that our electrons are used to do. Then we see here these hydrogen ions. We have this build-up, this concentration gradient, so that we can make use of it through this molecule. So we talk about chemiosmosis when we talk about this concentration gradient. And then this molecule here is our ATP synthase. This whole thing here in purple, kind of like a little turnstile. It's a mechanical action that the hydrogen ions moving through. It's used as a big enzyme molecule to join ADP and inorganic phosphate to make our ATP here. So we call this photo because it's driven by light, phosphorylation, adding phosphorus to our ADP. So these are important steps that are happening in our light-dependent reactions. And then, of course, we have that carrier molecule that's here, that's this guy, NADP. That picks up hydrogen, and it also picks up excited electrons from this photosystem. So these ones in this photosystem here went down an electron transport chain, and electrons from this one went straight to a carrier. Now, if an electron is lost from here, it gets replaced by this one. So this one comes along and replaces the one that's gone from here. Where does this one get replaced from? This one gets replaced by these electrons from the splitting of water. So again, a little more information. We need to recognize that actually, because they're doing slightly different jobs, we have 2 different photosystems. So photosystem 1, slightly counterintuitively, is actually this one. This is photosystem 1 over here. And that one releases photoactivated electrons. So our electrons here shown in this kind of orange pathway, they join up to our carrier molecule. If you see it in textbooks written with a number, that just indicates the main wavelength of light that's absorbed. Photosystem 2 is this one over here. That replenishes these electrons, and its excited electrons are used in that chain to drive all the hydrogen ions into the little space. And I've color coded them because this one is more of a sort of true green, and that one's a kind of more teal green as the main wavelength of light. And of course, we showed here water is the source of our electrons to replace them in photosystem 2. Now, this picture you've seen before, you've had this handout in class. So if you want to grab another copy and annotate along with me, you can do it again, or just watch and see how much you remember. So here we've got our photosystems 1 and 2. We've got a hydrogen pump, and we've got our ATP synthase enzyme there. So this is the same sort of diagram as before, just building it piece by piece. Light will come in or strike photosystem 2. Out goes an electron. It's excited. I've just shown it going up as a way to say it's elevated in energy. And then it's passed along these carrier molecules, which the energy is used to drive hydrogen ions being pumped into this thylakoid space. And that electron is going to go to photosystem 1 because at the same time, photosystem 1 is having an electron also excited. That one is going to the carrier molecule. To replace this electron from photosystem 2, we split water, photolysis. So we take some water and we generate oxygen gas, and some more hydrogen ions. So the combination of those hydrogen ions coming from water and being pumped across by our electron transport chain, we end up with a very high concentration of hydrogen ions in this very small space. The fact that this lumen is also quite small exaggerates that concentration gradient. And then they can pass through that ATP synthase molecule, and they can join this NADP here, that's the hydrogen, that's joined with the excited electron. And the energy in that enzyme can use then to make ATP. So they are our light-dependent reactions. Splitting water, generating ATP, and loading this carrier molecule here with hydrogen ions and electrons. So now we see how that goes with the Calvin cycle here, the light-independent stage. So this is centered around carbon fixation, with carbon dioxide coming in as an input, and of course our output is organic, mostly carbohydrates, sugars, but also then can link to other biochemical pathways to make amino acids and so on. And we return the products of the Calvin cycle, the reusable components. If I just remove that little bubble, that's going to stay there. I can't go back, apologies. We return our ADP and our NADP go back to the light stage so they get reused. So here we look at our stages of the Calvin cycle. Our first molecule is this RUBP. This is a 5-carbon molecule. That's what the grey sort of circles are showing you. It's not showing you the entire molecular structure, it gets too complex, but focusing on the number of carbons is important. And that's a common thing in an exam question that'll be annotated as being like 5C. So here we can see that our input of carbon dioxide is catalyzed by an enzyme. This is an important name to remember for an exam question, Rubisco. This does not require energy. That's also an exam question sometimes, asking which stages of the Calvin cycle need ATP. It is not the actual stage of carbon fixation here. That is just an enzyme catalyzed reaction. So we have here, and then we go to GP, or glycerate 3-phosphate. So we go to a 3-carbon molecule, and we can see that they've actually used multiples in this so that all the numbers work out. Now this is the stage where we have a massive input of energy. We have lots of ATP, so we return that ADP to the light-dependent stage, and we also unload this little carrier, kind of like our little bus driving our hydrogens around and our electrons. They hop off so that we can incorporate some hydrogen into this reaction here. Now, because we've got those hydrogen ions, this is a reduction process. Of our GP to TP. We've still got a 3-carbon molecule though. So we haven't changed the number of carbons, but we have changed hydrogens, and we have changed our oxidation state. Now at this stage, out of the amount that we've got here, 1/6 of it leaves to go and be incorporated into other larger organic compounds. And the rest is used to regenerate our starting molecule. And that's what makes it a cycle. So here we have the same molecule. We're still dealing with TP, so you don't actually need to write that again if you don't want to, because you've just got our triose phosphate here. But to get from 5 lots of a 3-carbon molecule to 3 lots of a 5-carbon molecule, there's some rearranging that has to happen, and that requires an input of energy. So we actually need some ATP here at our last step. So of our main stages here, we have fixation using an enzyme, and then we have reduction and ATP, and then we have only ATP to recombine our molecules to get back to our starting point in our cycle. Now, how these different chemical stages were discovered by Calvin and some of his associates were by using some algae in this little lollipop-shaped flask here. And what they did, they used, as with many of the biological experiments we see in our course, they used an isotope. So they were able to tag the molecules that were coming out by looking for what proportion of radioactivity they had by using this radioisotope carbon-14, which is heavier than our common isotope, which is carbon-12. So little algae are in here. The light was turned off to start with, but they made sure there was access to air and other nutrients that they would need for photosynthesis. And then here at the bottom, some books say ethanol, others say methanol. Either way, we had an alcohol here, or even hot alcohol to try and kill our algae as soon as this little valve was released here to take a sample. So we can see that the light is switched on and the timer started, and the algae could start undergoing photosynthesis. And after a few seconds, you could let out a small amount of the sample and put it in the hot alcohol to kill it, and then replace this flask down the bottom. So that every few seconds, you have like a frozen in time little sample of your algae to see the order in which all those compounds were made. So all those stages of the carbon cycle, the GP and the TP and the IUBP, they were looking to see which order they were assembled. They were trying to elucidate that biochemical pathway by looking for the relative amounts of each and what order they were made in.